So today I'm going to briefly introduce LabKey, and then we're really going to spend most of our time in a live demo. We've got a really fun fictional scientific scenario. We're going to outer space today, but the workflows that you see are going to be really similar to what real labs deal with every day. This just gives us a nice way to show how flexible LabKey LIMS is without getting too specific in any one industry. So before we jump into the demo, I want to spend a few minutes on who LabKey is and what our LIMS offers to teams. I'll also introduce myself. I'm Hannah Brackey. I'm the VP of product here at LabKey. Prior to working at LabKey, I was a customer in the lab working on translational medicine. And so I have a lot of experience in dealing with workflows and trying to map protocols to software that our labs are using. So a quick word about LabKey. We've been around for over 20 years, working across life sciences, environmental sciences, mining, agriculture, and more. And what that really means is that we've seen a lot of different types of lab workflows, both sort of in the regulated space and non-regulated space. And we design our software to really adapt how labs actually work rather than trying to force you into a really rigid process or requiring you to change your SOPs to fit the software. And today's demo is really aimed to reflect that. And so we'll have a look at that. We've got multiple sample types, different workflows, and different types of data that are all living together in one centralized system. What makes LabKey different is that we're not just a software vendor. We really see ourselves as a partner in science, which means we tend to spend the time necessary to really understand how your lab works, help you configure those workflows to fit your reality, and train you so you're able to do that yourself moving forward, and support you all the way through that adoption phase as well as ongoing maintenance, because no lab ever stays the same for long. You'll want to constantly evolve your processes within the LIMS to match what your science is doing. So at a high level, LabKey LIMS is a configurable low-code cloud-based platform that's really designed to be easy to use in real labs. So instead of forcing you into these hard-to-change workflows, it adapts to how your lab operates and continues to adapt as your science evolves. One of the biggest challenges we see is data spread across spreadsheets, different ELNs, instruments, and shared drives. LabKey can help connect that data so teams aren't constantly reconciling information by hand or spending time hunting for data that should already be accessible. And as labs grow or become more regulated, permissions, traceability, and audit readiness are already built into the system. They're not bolted on later when some QA requirement shows up multiple months or years down the road. So let's make this a little bit more concrete in terms of LabKey LIMS, because this really will map directly to what you're about to see within our system. It really starts with registration, being able to uniquely identify every sample or material that comes into the lab. And that's really how you can avoid mislabeling and loss as the volume of material increases within your team. From there, LabKey maintains that end-to-end traceability across people, instruments, locations, and handoffs. So you always know what a sample status is and its history. Labs also use LabKey to standardize repeatable workflows, preparation, testing, review, release, with clear handoffs and checkpoints without having to micromanage team members. And then all of the results are captured with full scientific context, metadata, methods, attachment, related records, so that data actually becomes usable later instead of just stored. And then that payoff is sort of the audit readiness piece. You don't have to reconstruct what happened. The record is already there when you need it, either for troubleshooting purposes, which we used to use in an R&D environment all the time, or when you're being reviewed by an auditor. And so today we'll look at each of these during the demo. So in today's demo, Research Institute has returned from this deep space mission to a planet called Xenon 9. They've brought back two categories of samples: environmental samples from different planetary sites and biological specimens from newly discovered organisms. The lab's goals here are actually pretty familiar, hopefully should be familiar to you. The goal is to sort of collect and process those environmental samples, analyze the composition, collect the biological samples, and then extract, sequence, and store that DNA. And so across all of this, they're really wanting to be able to answer any of the questions at any time. Where did this sample come from? How is it processed? And what data was generated on it? So today we're going to show you how that works in practice, and we're going to do it all inside sort of a single workflow job. This will follow that full protocol from collection, site registration through the sequencing results. And that's kind of what I have mapped out here. And so we'll look at and see how this protocol that this fake lab has transitioned into our live LabKey demo site. So this is LabKey's dashboard for our fictional project, Project Xenon. You can think of this as your lab's home base, a real-time snapshot of what's happening right now in the lab. Across the top, this works the way that you'd expect. You can search for sample sources and other records globally from here. You can also switch between projects and access any notifications or system settings. And the nice thing about this global search is that you really don't have to remember where anything lives. You can just search here and find what you need. And everything you're about to see lives within this Project Xenon folder. Folders within LabKey let you organize work by study, program, customer, or mission, depending on how you choose to partition it, while it's still using those same underlying system. So if you have multiple studies or teams, you don't end up with a bunch of different tools or even different tables. You just have these separate spaces that people have different access to. So on the dashboard here, you can see all of the samples within Project Xenome, what we currently have in the lab, and anything that's been processed. The point really isn't these exact numbers. It's really about getting that overview into understanding the shape of the work in the lab. And I've customized this as an administrator, and this would look very different, obviously, for the science within your organization as well. Down here, we have our active workflow jobs, what's in progress and what's due, and what might need attention. And as a lab manager, I can go in and select all of the active jobs rather than things that are just in my queue for something that's a bit more high level in terms of what the lab's working on. We also have the storage activities. So this allows me to see the boxes that are being added to on a daily basis. And the last thing is our built-in electronic laboratory notebook. And so I have 2 in progress notebooks. I would also be able to go in and see any notebooks I have for review here as well. So where we're going to start today is actually our workflow. And I'm going to view our webinar workflow. And this is coming from a template that I made that specifically has these tasks. And so if we look at each of these tasks, they map pretty closely to what our protocol had stated. We're registering the environmental and biological samples. We're processing those environmental samples into extracts. I'm also making sure that we analyze those extracts and then store them afterwards. We then process those biological samples and extract the DNA. We store the reserved DNA samples, and we're actually taking a subset of those to sequence and then store post-sequencing. At the very end, we have a QC review that will look through the entire process. As part of my workflow, I also have the ability to set who should be assigned the work. And so right now, this is assigned to me. I can also add folks to the notify list. And so they're getting up-to-date information about what's happening with this workflow. I can set priorities and have a start and due date. I can also add additional metadata like project codes or lot numbers or things like that that are useful to track moving forward. We can look at the samples tab for the samples that are already part of this workflow job. And so we have 8 biological samples and 8 environmental samples. For each of these different sample types, again, me as an administrator has really configured the language here on what these samples are called, as well as all of the fields that are relevant to this sample type. For each of these samples, we have a system generated unique identifier, and we're pulling through actually the organism name as those identifiers. I'm able to see the organism as well as the collection site from the biological samples. If I click into one of these individually, I can see that metadata again, but the most important thing is actually being able to see this lineage diagram. And so we'll come back to this lineage diagram as we process our workflow, as we have more extracts or DNA samples that are getting generated from it. This lineage diagram is interactive, and so I can double click and actually look at this organism and see what else has been generated from this organism or even from this collection site. If I go back to that same sample, the other thing that I want to note here is I can see the aliquots, assays, and workflow jobs that the sample has been assigned to. But importantly, I can also see the timeline. So we can see when the sample was registered and then when it was added to this workflow job. So again, we'll come back to this timeline over and over as we execute the work today to see how the audit record for this specific sample grows over time. So I'm going to go back to my workflow job. We can also see the assay tab. And right now, because we haven't executed any work, this assay tab is empty. But as we start to go through our tasks, those assays will come through. Again, these are assays that I defined as an administrator. I also indicated, what are the fields that we're capturing on these assays. So let's actually go through it and start to execute the work. So this first task of registering the biological and environmental samples, I sort of preloaded our data already. So I've already done this. But what this task also wants me to do is make sure that we're updating these sample statuses to registered. So if we look at those samples again, we don't have a status associated with them right now. So I'm going to go back to tasks and I'm going to update that status. I can always provide a reason for update, and this gets captured in my audit log as well. Those samples are now updated to registered. The next piece of this is making sure that we're adding them to storage. So we're going to collect these samples, register them, and put them in storage for when we get back to the main site for processing. So I've got a CryoBay Alpha here that I'm going to add these to. I'm able to choose sort of the positions that I want and choose the order that I want. Maybe one of the things that I want to do is sort of physically separate the environmental and biological samples from each other. I'm able to do that. Once I've added these to storage, I'll complete this task, and this is now done. If there were other team members assigned these next tasks, they would be notified now that they have work to do within the system. So this processing step is about processing the environmental samples and putting them into extract. So we've made it back to our main lab. What we want to do now is extract these environmental samples. One of the things that I'm able to do here is either derive these manually or import a file. I'm going to do it manually because it's easier to see. One of the things that we've set up as part of the workflow template is being able to say we want one derivative for every environmental sample. I also, once I've derived these environmental extracts, I want to update them to being processed and remove them from storage at the same time. So this really minimizes the amount of manual effort that I have to go in and do within the system, which really reflects kind of the reality of what we have in our lab. So I'm going to go to this sample grid. I have data here. Again, I could have uploaded this via file, but I've got it sort of copied and pasted. The system is going to generate these sample IDs for me. We're associating each extract with an environmental sample. And so I'm going to create these samples, and not only are they going to be generated and associated with the samples, but these extracts are also going to be added to the job. So we can see here that our sample list has now grown. We've got 8 environmental extracts. If we go into one of these, we'll take a look at the lineage. And now we can see we've got this extract associated from our sample. If we go to the collection site, we can see the much larger group of samples that have been generated. If we look at the sample timeline here, we're also going to see that it was registered and added to that workflow job. So I'm going to complete this task. And the next thing that we want to do then is analyze the environmental extracts. And here I have importing that environmental analysis data. This, again, is something I can do with a file. I could do it more automatically. For the sake of sort of the demo, we're going to do it manually so you can see what this looks like. I'm going to choose to import the data. Got some information here. I can fill out kind of the date of my assay and the assay type that I'm doing. I can put in additional comments and set an ID if I want to. I'll import this assay data. And now we've got our assay data associated with these samples as part of the workflow job. The next thing this task wants me to do is also indicate that these environmental extracts have been analyzed. So I'm going to update the status here and then add them to storage. And so we'll now sort of have these environmental extracts that we want to add them to storage. And so here, they've already been added to storage. And so the task is essentially complete, and what it looks like when I'm doing a live demo. So we'll complete this task. And then we can move on. So I can continue to sort of process these biological samples and store them and remove from storage. And the process is going to be fairly similar to our environmental samples. When the job is complete, it's actually going to look more like this example job where we have multiple different assays associated with samples, as well as our sample listing that has both our primary biological and environmental samples, but then also the derivatives of DNA and environmental extracts. If we look at one of these lineage diagrams again, we'll actually be able to see kind of what this looks like in practice when everything has been completed and these samples have been processed. The timeline will also demonstrate that all of the different things that have happened to the sample. When I select on one of these things of an update, we're able to see exactly what changed during that update as well. If I go back to the workflow job, the other thing that I can do here is analyze my assay data. So we have some kind of built-in analysis here that allows us to see the result status, but I could also build a chart here to be able to look at, let's say, sort of my mineral index by collection site. And that gives me kind of a quick overview of how my assay performed, but then B, an indication by collection site of what this sort of index looks like. The last thing that I'll represent here today is our ELN component. And so quickly, I'll just go into the biological sample analysis. And I've been able to generate a kind of plain text notebook that's associated with the data within the system. And so as we kind of scroll here, you can see that I have different samples that are associated with samples within the system. We're capturing those samples over here. I'm able to go through and interrogate those samples even more, or submit this for review and get sort of formal sign off on the notebook as well. And so with that, we'll go back to the slides here and kind of wrap up. LabKey LIMS is really designed to have these lab managed workflows. We want all of the labs to be able to manage these workflows on their own and configure them without code. That traceability that we saw as part of the lineage, as well as bringing all of those samples and assay data into the workflow, is captured as the work is performed. It's not something that needs to happen later or as an artifact after the fact. As you hopefully have been able to see, it has a really structured, adaptable data model. So if you don't have biological samples and DNA samples or these environmental samples, you are able to decide what is the language your lab is using, as well as what those fields are that are relevant to you. LabKey is also AI ready and enabled via an MCP, so you can take the data here and be able to interrogate it in your LLM of choice and really take the analysis much further as needed. And so as the last kind of wrap up here, one thing that you can expect if this looks interesting to you, we really like to take people on this journey of a guided evaluation. And this has us go through sort of these three main steps: being able to discover what your needs are in your laboratory and do a deep dive into the demo. This is a very quick overview today. After that, we'll really actually work with you to configure the system in ways that make sense for your lab and demonstrate how that might work. And that allows everybody to get hands-on experience with the system before a purchase decision. And then lastly, we'll have that trial. And so that's really allowing you to go in and test it out yourself, and allows you to ask questions and make modifications as it goes on. So keep an eye out for your email for a link to schedule that initial discovery call, and I look forward to hearing from you guys. If you have any questions, it looks like there's some questions already in the chat, so I'm just going to read through those quickly and try and answer as many as I can here. Yeah, so the first question here is the ability to have a paper lab notebook as part of the system as well, or a digital lab notebook, I guess, that has that reference too. And so hopefully you were able to see that as part of the ELN, where you can actually make that reference. And let me see if I can go in and show you the site again. And if I go into an individual sample, for example, and take a look at what we have here. So if this sample had been referenced in a notebook, we would have that notebook reference that would show up here. And then, yes, it would also, it wouldn't show which page because we're a browser-based system. We don't have sort of a formal pagination like that, but you are able to view the reference within the system and go to that first reference in the ELN. Can LabKey be configured into different kinds? So 96 deep wells, 384. Yes. So our storage accommodates multiple different types of containers for storage, such as 96 deep well plates or 384 well plates. One of the things that we have in our LabKey biologics system that we're very excited about pulling into our LIMS eventually is our plate management. So if you're actually executing assays in plates, you're able to do that as part of that system. Sometime also be able to do that within our LIMS. How does barcode scanning integrate into the steps? So our system does allow you to both generate barcodes for samples and also scan them. So if I am working with samples and scanning them, I would be able to find them by ID or something like that. But if I'm within a workflow job, one of the things that I would be able to do is register them in that grid by scanning the samples. And so that would be one method of sort of tracking it, or tracking, being able to scan samples within the grid directly as well. So if we go into this in progress and I have my drive task, I would be able to scan those sample IDs down here as well. Background calculations. Yes, so you're able to add calculations within our system, both at the sample level as well as the assay level, and track repeats across different workflow runs. Yes, so there's definitely kind of the notion of being able to have multiple assay results for any given sample. And so you can see, we might have a review status here. I have customers that have configured these to be failures, which will then sort of remark that sample for, a new DNA extraction or something like that, that will help kind of kick them out of the workflow that they're in and push them into the queue for a new workflow. Okay. So with that, if there are other questions that folks may have, please feel free to either schedule some time to talk to me about it or send me an email. And I look forward to kind of working with you to help map your protocols to LabKey LIMS workflow.